Hi, my name is Taryn Collins, and my presentation is on an article called The Impact of Integrase Inhibition Compared with Non-Nucleoside Inhibition on HIV Reservoirs in Lymphoid Tissues. The primary researcher for this article is Dr. Megan Rothenberger, and the article was originally published in the Journal of Acquired Immune Deficiency Syndromes. While there are several types of antiretroviral drugs available to treat HIV, the two types focused on in this study were integrase inhibitors and non-nucleoside reverse transcriptase inhibitors. Integrase inhibitors work at step 4, right here, and prevent HIV DNA from becoming part of the host DNA. Reverse transcriptase inhibitors work at step 3, and the one used in this study works to bind to reverse transcriptase to prevent it from making HIV DNA. The two specific drugs examined in this study were efavirenz, which is a non-nucleoside reverse transcriptase inhibitor, and raltegravir, which is an integrase inhibitor. The authors of this study wanted to investigate which of these two drugs works better. The scientists quantified this by measuring how much of the drug was still in the body after different periods of time, with the reasoning being that a higher concentration of the drug would treat HIV better than a lower concentration of another drug. In particular, scientists focused on results from the lymph nodes because the lymph system is greatly impacted by HIV, and many HIV-infected cells can be found in the lymph nodes. For this experiment, the researchers recruited 11 participants to the study who had never taken HIV treatment before and were HIV positive. These participants were randomly given raltegravir or efavirenz, along with 2 other antiretroviral drugs. Participants were given 3 drugs at a time because that is the standard of care for HIV. Blood and tissue samples were taken over the course of 6 months. These samples were then analyzed to measure viral load, infected cells, and the concentration of the medication. Figure 1 shows those measurements from the participants over time. In B, you can see that the raltegravir and efavirenz lines mostly overlap each other. This graph shows the concentration of cells that are infected with HIV in the lymph nodes. Lymph nodes have a high concentration of the cells that are targets of HIV and are vital for fighting off infection. Visually, this graph suggests that there is very little difference in the effects of the drugs in changing the concentration of HIV in lymph node tissue. This table shows the differences observed between the results in Figure 1. The researchers used a p-value to decide how likely it was that the results could happen from chance based on the sample size. A p-value of less than 0.05 is considered statistically significant. Anything greater than 0.05 means that the result could have happened without the change in the variable. The p-value for graph B of Figure 1 is 0.365, which means there is no significant difference in the results for efavirenz and raltegravir. Figure 2 compared the concentration of the drug found in different parts of the body to a clinical threshold. In this case, the clinical threshold is the concentration of a drug that is expected to be effective in treatment, with 1 representing the threshold. In the lymph tissue, the researchers found values from both efavirenz and raltegravir that were below the clinical threshold. However, efavirenz was much closer to the clinical threshold than raltegravir. As you can see, efavirenz showed a concentration of 0.95 compared to the 1 of the clinical threshold, and raltegravir showed a concentration of 0.03 in the lymph node compared to the 1 of the clinical threshold. Table 2 gives the specific values from Figure 2. The efavirenz is being compared to a clinical threshold of 700 nanograms per milliliter, and the raltegravir is being compared to a clinical threshold of 60 nanograms per milliliter. Once again, both values are below the clinical thresholds in the lymph nodes, though efavirenz is much closer to the clinical value than raltegravir is. The hypothesis for this experiment was that raltegravir, the integrase inhibitor, would have a greater concentration in the lymph nodes than efavirenz, the non-nucleoside reverse transcriptase inhibitor. In the experiment, however, efavirenz performed better than raltegravir. However, neither drug met the clinical threshold to predict efficacy against HIV. One limitation of this study was the number of participants. With only 11 participants, it is difficult to generalize the results of this experiment to other people. If this study were performed with more participants, it would provide more data points to give a fuller picture of how concentrated raltegravir and efavirenz are in human lymph nodes. And here's the work cited with information about how to access that article with the DOI, in case you would like to read the article in its entirety.